want to talk a little bit about the plague bacteria. You might have heard about the Black Death in Europe, and that was because of the plague. And the plague was first isolated by a French bacteriologist by the name of Andrew Yersin. So this was first isolated by this, or it was studied by this French bacteriologist. And his name is Andrew Yersin. Andrew Yersin was the French bacteriologist who studied extensively this bacteria, and in his honor, it was called as Yersinia pestis. It's one of the gram-negative bacteria. It's a rod-shaped bacteria, and when you stain this bacteria, it gives the typical appearance of a safety pin under the microscope. And that's one of the unique features. You can identify Yersinia because of the typical unusual bipolar staining with a safety pin appearance. That's the type when you look at under the microscope following gram stain. There's a bipolar, that means on both the ends, because it's a gram-negative rods, on both the ends of the rod-shaped structure it will be stained, giving the typical appearance of a safety pin. It's one of the zoonotic pathogens. Zoonotic pathogen number one, like anthrax, they are also prevalent, common amongst animals, but humans are also susceptible to it. So this is one of the zoonotic pathogen. It's pretty much common amongst rodents, squirrels, prairie dogs, and that's because they are present in one of these smaller animals. It is impossible. It is next to impossible to eliminate this pathogen. It's because they are present in this thing, they are almost virtually impossible to eradicate or to eliminate this pathogen. So there is no eradication or elimination. The transmission of this pathogen, it is often transmitted.
So just to clarify, zoonotic means that it will also cause symptoms in the other?
It may or may not cause. Some of the pathogens, because there is sort of what we call it as a balanced pathogenicity. When they are present in animals, it may not harm the animal. It will stay there, possibly a fertile reservoir. But when it jumps to human beings, it causes all the harm.
Okay.
But zoonotic pathogen means rabies is a classical example. If you forget about all the things, rabies is a classical example of zoonotic pathogen. Okay. Now, this pathogen, its transmission is via this fleas. The flea is the one when they try to suck some blood from an infected vector, and then when it tries to suck the blood to human beings, that's the time it transmits this Yersinia pestis into the human circulatory system. So this is the one which is the transmission agent. It's mainly because of the bite of the vector. This is the flea, an invertebrate that transmitted to human being from animal to human. So there is a direct animal to human contact or human to human contact. That's how the spread is maintained by this for this pathogen. So there could be direct animal or human contact, and spread is commonly observed with this pathogen. And that I'm going to catch up in the next slide. What do I mean by this? Human to human contact and animal to human contact. Human to human contact is also observed among medical personnel. If one medical personnel is treating patients of the plague, and if he is not careful, he might be contaminated, and then he will contaminate some other patients. So it's a kind of a drag, because medical personnel, they can transmit some of the pathogen to the patients if they get infected. oftentimes, this pathogen, oftentimes, this pathogen, once it is recognized and once it is phagocytosed by the defense cell, it can still survive in the phagocytes. So it may be recognized by the macrophage, taken inside the cell, inside the macrophages and some of the defense cells, like the macrophage and the neutrophil, but it can still survive within the phagocytes, and once it is inside, the bacteria multiplies, then travels via the lymphatic system to the lymph nodes. And when it gets onto the lymph nodes, your lymph nodes get swollen, and one of the type of the plague, or one of the manifestations of the plague, is the bubonic plague, when you have a swollen lymph nodes, specifically near the armpit. And we are going to catch it up in the next one, when we talk about the bubonic plague. So once inside, the bacteria multiplies and then multiplies within the server, within the phagocyte. And what I mean is, when I say once it is inside the body. Once it is inside the body, it is able to multiply. It is taken up to the lymph nodes, and that's when it also again encounters some of the phagocytes, and the lymph nodes get swollen. There's high fever, chills, and shock associated. The high fever is because the defense system is trying to get rid of the bacteria once the bacteria is in the bloodstream. And that's one of the reasons. They can be identified and grouped as the bubonic plague, the septicemic plague, and the pneumonic plague. So these are the three forms. Of the three forms, the most lethal one are the pneumonic plague. The pneumonic plague is the most lethal one, and the three forms of the plague are the bubonic, septicemic, and the pneumonic. These two, the A and the B, are the one which are transmitted by the fleas. The bite of the flea is able to transmit the bubonic plague and the septicemic plague to human being. Okay? The pneumonic plague is mainly person to person. The pneumonic plague is mainly person to person as an aerosolized particle during coughing, sneezing, talking, and etc. that way. This is communicable. That means it can be transmitted from one individual to another individual. And at the same time, this is the one which is extremely fatal. This is the one which is fatal compared to the other two. Bubonic plague, you have specifically of the armpits and the groin. This is where you see this person's. This is where you see. This is the place where you have the lymph nodes under the armpit is swollen. Do you see it swollen? And that's because of this bubonic plague. You have the swollen lymph nodes. The septicemic plague is when the bacteria enters the bloodstream, it may cause a shock-like syndrome and the DIC. So when the bacteria gets into the bloodstream and causes some of the shock-like syndrome and DIC, that's when it is called as a septicemic plague. Septicemia is a clinical term which means the presence of bacteria in the bloodstream.
3 different bacteria, or are these all?
Same bacteria. Same bacteria. It depends on how we have acquired it.
Okay.
Depends on how you acquire it. If you acquire it from one person to another person by coughing, as an aerosolized particle, it will manifest as a pneumonic plague. But if you get, yes, oh hi, oh. Septicemia is a clinical term which means the presence of bacteria in the bloodstream.
So you could get all 3.
You can get all
It travels around in the appropriate
But oftentimes, septicemic plague, like septicemic and the bubonic, they are, kind of not good. But then the pneumonic one is a really lethal one. It can cause a lot of complications, when it is present in the lungs. Okay. Virulence of this bacteria is because of many factors which are encoded either by the plasmid DNA or the chromosomal DNA, which I'm going to catch up in the next slide. And the treatment and the prophylaxis is some of the broad spectrum antibiotics are effective in killing this bacteria. So some of the broad spectrum antibiotics are effective. There is no resistant, drug-resistant strains have not developed yet, so we are kind of a little bit in a better shape. And lastly, your bet is to control the rat and the fleas. One thing to remember, sometimes back we had some of those, this strike by the waste management folks. And one of the biggest concern for the public health official was there might be an outbreak of the plague. That was the biggest concern, that there will be an outbreak of plague in the East Bay because the waste management folks, they were on strike, and we thought that this is going to be the boom time for rats because most of the household garbage was outside. The bins were overflown, the covers were not there, and the rat population, once it explodes, along with the rat population, there will be a lot of fleas. And those fleas will tend to bite human beings, and some of those rodents might be infected, and there will be an explosion of plague epidemic. Oftentimes we hear about plague epidemic. It's not that problem in developed countries, but sometimes we hear it erupts off and on in New York area, and in some of the other parts of the globe it is endemic in certain parts. But most areas, the public health department tries to take care, so there's no outbreak of plague. Is that clear? Huh? I know. So possibly that one, because, and that's one of the reasons you always need to cover your garbage bin, because if you keep it open, that will also cause the rat population to be there, and then there's an increased risk of getting the plague bacteria. A vaccine is available for some of the high-risk individuals. These are the folks who either work in the zoos or they deal with some of these exotic animals. They are more prone, so there is a vaccine available. But the common individuals, the common folks, they are not being vaccinated. Yes?
Professor, is this the black plague that targeted Europe a long time ago? Why back then did it kill so many people, but now it's not a huge problem if it's the same?
Because that time we didn't even know how to control it. We didn't know even how to control it, and some of those treatment options, we were not sure what is going on. That's one of the reasons why it killed so many people, like the Spanish flu also killed. But now we know what is the reason why the Spanish flu killed so many people at that time. The same thing, and it's kind of a similar approach. Yes?
And how does it do that to the skin?
How does it do that? It's because of tissue necrosis. It's tissue necrosis, and oftentimes some of the Clostridium species, they tend to do this.
Is that from a toxin?
Yeah, the Clostridium toxin. Now, the next part we talk about is, here's the one, how it is transmitted. There are some of those, what we call it as an amplifying host. Amplifying hosts are the deer, cats, dogs, and some of those, not the rodent family of animal, but some of the things which are away from the rodent family. That increases, it tends to amplify. It tends to increase the number of bacteria. That's why they are called as amplifying hosts. And here it is shown, so the natural endemic reservoir are the rodents, and it can come in, when humans come in direct contact with the rodents or by the bite of the flea, they can get this, they can get the plague. It can also be from person to person as aerosolized or airborne transmission. It can be from person to person. It can be from the rodent to a person by direct contact. If the rodent comes in direct contact, it can transmit it. Or it could be by the bite of the flea. So these are some of the ways by which the bacteria can be transmitted to human beings. The next slide and the last slide, are the plasmid which are present and that carries the virulence factor of plague. There are 3 plasmids which are present, and that the 3 plasmids are the one that they carry the virulence factor of the bacteria. It may sound pretty gross here, but then I'll be short and sweet and to the point. You don't have to know everything. There's a small size plasmid, there's a mid-sized plasmid, and there's a large size plasmid within each of those pathogenic strains of Yersinia. The small size plasmid codes for a protein, and what that protein does, it dissolves the blood clot and it prevents fibrin clot formation. This is something, a response of the human defense system. Whenever some non-self pathogen gets into the bloodstream, your first response by the defense system is to contain the spread of the pathogen. And the way it contains it is, it triggers the clotting formation. It triggers that fibrin clot formation. So once the bacteria is inside and the fibrin clot has been formed, that fibrin clot will try to trap the bacteria and it would prevent its spread within the body. Does that make sense to everyone? Okay? So the small size plasmid codes for a protein, and what that protein does, it acts to dissolve these fibrin clots. And also it would prevent the clot formation in the first place. If there's any clot formation due to the action of the defense system, it will try its best to dissolve it or to prevent the clot formation. Okay? The mid-sized code for a protein which is called as YOPS. YOPS stands for the Yersinia outer membrane protein. Yersinia outer membrane protein is the YOPS. And the YOPS, what it does, it targets the cytoskeleton of the defense system. So the defense cell is not able to engulf and eliminate the pathogen. So it targets the cytoskeleton of the defense cells. So it interferes with the phagocytosis signaling as well as the elimination of the pathogen. Okay? So it interferes with the signaling pathway, and then it's not able to, because whenever we talk about the engulfing, there is the cytoskeletal rearrangement, and that's how it is able to engulf the pathogen. And that signaling and that cytoskeletal element, actin cytoskeleton, is messed up by the mid-sized plasmid. The outermost, the largest one, it codes for a protein called as a F1 protein. F1 stands for the fraction number 1. So F1 is the fraction 1. That's the F1 protein. And this fraction 1 becomes a part of the capsule. It is antiphagocytic, and it stops the bacteria from the phagocytosis process. So these are the 3 main reasons. These are the 3 primary reasons why the bacteria is pathogenic, and these are some of the virulence factors which are encoded by the 3 plasmids. Interfering with the clotting process, interfering with the actin cytoskeleton and signaling, and the third one is a component of the capsule which is strongly antiphagocytic. The fraction 1 is also the one which is used for the vaccine preparation. You should know this one. It's called as a fraction 1, but this is the one it is also used in the vaccine preparation. Most vaccine of the plague bacteria, they have the F1 component as a vaccine. All right? Now, the virulence is mainly because of the adhesins. You know what is the role of the adhesins. There is a type of secretory system called as a type 3 secretory system. The type 3 secretory system is also referred to as injectosomes. And it's pretty much the type 3 secretory system is a whole complex. I don't think I've done a secretory. Did I do the injectosomes in this class? No. Yeah. Not yet. I've not discussed it. This type 3 secretory system is also called as injectosomes. Injectosome. And it is pretty much like a flagella type thing. And it's called as injectosome in the literal sense. It acts like a hypodermic syringe. That's why it is called as an injectosome. It literally works like a hypodermic syringe. What the bacteria does is it will get closer to the targeted cell and use this injectosome to introduce some of this component in the targeted cells. Just like you push it, you inject somebody with some of the medication. The same thing, the injectosomes, they use to secrete, they use to introduce some of those antiphagocytic proteins. Like the part of Yopes can be is one of the antiphagocytic proteins. So it introduces those into the host cell, and once those are introduced into the host cell, which could be the cells of the defense system, it could be the macrophages, it could be the neutrophil, it could be those dendritic cells of the immune system, and that would stop. It would fool those immune system cells. They will not elicit a response, an immune response towards this bacteria. It's a pretty evolved bacteria. Look at it, when you want to be evil, if you want to screw someone, you can really come up with ways to screw that person. Okay? John, look at this guy. He's gone a little bit too far, It uses the injectosome capsule. It uses the adhesin to stick onto the cells. It uses the capsule as an antiphagocytic factor. On top of it, it introduces some of those things into those cells of the immune system. Those dendritic cells, the macrophages, and the neutrophil, those are the front line of the immune system, and it paralyzes these cells. Does that make sense? Folks, I'm giving up on the tuberculosis right now. The next one is the tuberculosis. Oh, sorry. I hear you. Plague is grouped as one of the re-emerging disease. It's one of the re-emerging disease, and I put a question mark, what is a re-emerging disease? Tuberculosis is also a re-emerging disease. So this re-emerging disease, specifically the re-emerging disease has been defined in the tuberculosis slide over here, Tuberculosis is a deadly re-emerging disease. These are the diseases which previously it was a pain. It was a big issue for the public health department and the public health problem. The incidence had decreased over the course of time, but again, they're coming back. That is what is called a re-emerging infectious disease. We had those problems earlier. We tried to control them. We have tried to eliminate them. We have decreased their chance, decreased their number, but now we see again a surge in those diseases and the pathogens. Tuberculosis is one of those worst of the re-emerging infectious disease. One of the reasons tuberculosis is coming as a deadly re-emerging is because of the increased number of HIV and AIDS patients. It's one of the opportunist infection. So we have to again, go back and find out. We have to literally go and then, do the chain of event to find out what can we do to stop the tuberculosis. It's one of the opportunist infection of HIV and AIDS individual, and because of an increased number of AIDS individual, that's one of the reasons why tuberculosis is a re-emerging disease of the developed nations. Plague is also one of the re-emerging infectious disease, and a few reasons why it is a re-emergent is because of its inherent communicability. It's not a non-communicable disease. It's not a non-communicable disease like tetanus. You have the tetanus, you suffer, you won't be able to transmit to the person who you hate the most or love the most. You won't be able to transmit. Unlike herpes, you can transmit to any person. But this you won't. So this one is you can, it has an inbuilt communicability. It can be transmitted as aerosolized particle easily. At the same time, there is a quick spread. If one person has the plague, it can be transmitted easily as aerosolized particle to a number of people, although the extent or the efficiency is not the same as the measles virus, but still it can be transmitted with much more ease compared to some of the other pathogens. Rapid clinical course. The bacteria, you don't have to, it gets inside, there is a rapid clinical action. The course of the person getting the infection and getting to all those signs and symptoms is very quick. So by the time you realize it, well, it's already done. Yes.
So why is it re-emerging then if none of these have really changed over time? It's not becoming more resistant to drugs. Why is it still considered emerging?
It's a re-emerging because more than likely it's called a re-emerging is because at one point we tried to control. We still have control it, For most of the developed countries it has been virtually eliminated. It cannot be eradicated because it's a zoonotic pathogen. It just cannot be eradicated completely, but it was under control. But again, now it is getting out of control because of another thing is urbanization. We try to some of the wooded areas because population expansion, we are basically crawling into that areas. Those rats and those rodent population coming in contact with human beings, that's why we're getting a re-emerging. Okay. Rapid clinical course and a high mortality if left untreated. So these are some of those reasons why the bacteria has been reclassified as a re-emerging pathogen along with tuberculosis. Tuberculosis, Streptococcus pyogenes, these are some of the re-emerging pathogens. There's a whole range of re-emerging pathogens which we discuss it in the infectious diseases class, but then I don't have the time to go over all of those, but we'll talk about a few of those. Does that make sense, folks? I need to stop it here. Is that